I want to begin by thanking the organizers. As Jeremy said, this is a fantastic opportunity for somebody like myself. I'm very much an experimentalist to have the opportunity to collide with people who have different but very complementary sets of expertise. So, as we all know, the challenge of connecting the activity of neural circuits to behavior requires collecting and making sense of a great deal of data. And what I'm going to gear my talk toward tonight is the behavioral end of this data collection, the behavioral end of this effort. So, as a postdoc in Tom Jessel's lab at Columbia, I've been exploring the neural basis of skilled movements. And so why study skilled movements? Well, movement is our means of interacting with the world. It's how we translate our intents and our desires into action. And I think one of the most impressive accomplishments of the mammalian motor system is our ability to move our limb very rapidly and precisely towards a target. And so, why study these skilled reaching movements, these skilled forelimb movements? I tell you, the main reason that I study it is because when I see a movement like this, this is my reaction. It's remarkable when you think about it. He's able to coordinate the contraction of dozens of muscles in his limb to bring his hand, or a tool like these chopsticks in this case, to a very specific point in space. So what I've been trying to do in the Jessel lab is use the genetic accessibility of mice to access discrete neural circuits in the spinal cord and the brain and try to identify the ways in which they contribute to these skilled forelimb movements. So of course the first challenge is to figure out if mice can actually do these kinds of movements. And luckily I didn't start from scratch here. Ian Wishaw and his colleagues have done a great deal of work showing that rodents can actually perform these movements very precisely and very rapidly. And there's a great deal of conservation in the structure of these limb movements between rodents and primates. So this suggests that if we can explore the neural basis of these movements in mice, then we might get insight into evolutionarily conserved features of mammalian motor behavior. So I think one of the challenges that I wanted to address is to try to develop an unbiased quantitative assay of these behaviors in mice. So I trained mice to reach for a food pellet that's located on the outside of a clear wall. And I used two high-speed, high-resolution cameras to capture these movements and track the location of an infrared reflective marker that I affixed to the back of the right paw. And so really what this let me do is reconstruct these movements in 3 dimensions and analyze various aspects of the kinematics of these behaviors. So I want to show you a movie of one of these reaches. Now the movie I'm about to show you is slowed down to about 6% real time. These movements normally take 200 or 300 milliseconds in a mouse. So you can see this is a successful reach. It's pretty dexterous. He's able to reach out, grab the pellet, and retrieve it. And what I'm plotting here is a trajectory of the paw in 3 dimensions. We can look at many aspects of the kinematics, so you can collect a great deal of data about these movements. Here I'm just showing the velocity of the paw with relation to the distance to the target. And of course we can look across many reaches, some successful, some unsuccessful. We can look across mice. And what we find, this is actually a very, very stereotyped behavior in mice, at least mice that haven't been manipulated in one way or another. So what I want to do tonight is describe 2 brief projects where we're able to apply this high-resolution behavioral assay to the investigation of the contribution of specific neural circuits to these behaviors. So, well, what do we know about a reaching movement? We know that the movement's not ballistic. What that means is it's not a single burst of motor neuron activity and motor output that brings the limb precisely to the target, but rather the motor output is tweaked and updated very rapidly throughout the course of the behavior. And this allows this mouse to refine its trajectory throughout the course of the behavior and get precisely to the target. So this indicates, in addition to the motor commands that generate motor output, there's some mechanism of feedback, something that informs us about the outcome of our behavior and allows us to correct the trajectory of these movements online. So I think the simplest possible schematic that could help explain this is something like this. So of course we generate motor plans in our brain, probably in our cerebral cortex. These plans are translated into commands that are conveyed down to the spinal cord. They eventually elicit motor neuron activity and muscle contraction. And of course when we move, we generate feedback from the periphery, proprioceptive feedback from our muscles that tell us about the position of our limb in space, visual feedback. And this peripheral feedback, this sensory feedback, is channeled back into the central nervous system, provides information about the outcome of the behavior, and in principle allows us to correct our movement when a mistake is detected. But there's at least one major limitation to this simple scheme, and it's that peripheral feedback is inherently temporally delayed in its trek back into the central nervous system. It takes time for these sensory signals to get back to the relevant recipient centers. And these temporal delays in peripheral feedback introduce at least 2 challenges that the motor system has to solve. Now the first challenge is, how do you maintain stability when you move? So it's a well-known feature of a feedback-driven system that the system can become unstable when that feedback is delayed. So an intuitive example of that is a thermostat that is unstable. Essentially it oscillates between heating and cooling because of the delayed feedback of the changing room temperature. So a straightforward way to introduce stability into the system is to reduce the gain of that feedback, reduce the impact that, for example, sensing that the room is too cold, reduce how much heating occurs, and so you very gradually and incrementally heat until you get to the desired temperature and you no longer oscillate. So this raises the question of whether our motor system, whether our spinal cord in particular, has a similar kind of sensory gain control mechanism that can limit the impact that this delayed sensory feedback has on motor output. Now, a second question, a separate question, is that even if you can maintain stability in the system, how do you make these very rapid corrections? When he's reaching towards that fly with the chopsticks, he can make these corrections incredibly rapidly. And so this suggests there's a more rapid source of feedback that's not quite as delayed, that allows us to assess the outcome of our motor commands and make corrections when we need to. And one idea that's been around for a long time in the field is that perhaps motor command pathways branch off and convey copies of these commands to the cerebellum. And so the cerebellum is thought to take these internal copies of these motor commands and essentially generate online predictions of the outcome of those commands. And in principle, what that allows us to do is predict the outcome of these commands and make corrections when we need to without having to wait for this delayed sensory feedback to arrive. But these ideas of sensory gain control and internal copies in the spinal cord have remained largely theoretical. And the main reason is because of the lack of experimental accessibility to putative gating and copy pathways. So for that reason, I've been trying to use the genetic accessibility of mice to try to gain access to specific neural circuits that we think might mediate these two functions. So as many of you know, different types of neurons express different molecules. And this is especially well understood in the ventral spinal cord where the motor circuits of the spinal cord reside. And so there are different classes of interneurons that each regulate motor neuron activity in their own way. And importantly, these different classes have different molecular profiles. And what this allows us to do is essentially test questions of causality. We can get access to these neurons specifically, one class at a time, and we can manipulate their activity. We can activate them, we can inactivate them, we can remove them from the circuit completely, and we can actually quantify the contributions that these neurons have to these reaching behaviors. So in the first set of experiments, we wanted to explore a potential sensory gating mechanism. And we've long known that in the spinal cord there's a special and specific class of inhibitory neurons. I'm not going to go into too much detail, but essentially these neurons are called presynaptic inhibitory neurons. And what's special about these neurons is they selectively inhibit incoming sensory information. And they're distinguished from the far more abundant postsynaptic inhibitory or GABA post neurons that are present throughout the spinal cord that form these postsynaptic connections, for example, onto motor neurons and to other interneurons, and therefore have a more general impact on the excitability of these circuits. So these GABA preneurons are very specific and only innervate these incoming sensory afferents. So the question is, well, do these neurons actually mediate sensory gain control, and what is their contribution to forelimb movement? So I collaborated with Andrew Fink, a former graduate student in the lab, and Andrew identified a way to access these neurons, and he demonstrated that they actually do reduce the gain of incoming sensory feedback. So we teamed up and figured out a way to actually selectively access and acutely eliminate these neurons in the adult mouse. So again, not going into too much detail, but we can combine some genetic tools with some viral tools to express a toxin receptor only in these neurons. And so here, the green axons labeled here are coming from these GABA preneurons, and then when we introduce the toxin, we can eliminate these neurons very quickly. So what happens to a reaching behavior? So you can immediately see this mouse has a lot of trouble. You can see there's this characteristic undulation or oscillation of the limb as he approaches the target. And it's really a very robust phenotype. So here in red is after we've eliminated the neurons. This is a trajectory in space, and I think these oscillations are even more apparent in this plot of the velocity across multiple reaches. So every time the velocity of the paw drops below 0, that's a reversal in the direction of the paw. So the paw is moving forward and backward again and again on the way to the target. Now, in data I don't have time to show you today, we actually found that these are very, very stereotyped oscillations. Across mice, they all seem to oscillate right at 20 hertz. They have this very characteristic decay time. And so what we wanted to know is, well, what's the simplest possible explanation for these very stereotyped oscillations? So we teamed up with Larry Abbott, who's going to be speaking here later. Larry's up at Columbia. And Larry developed this really nice model of the limb with a single joint that's driven by these flexor and extensor torques. And what Larry was able to do is change the strength of the incoming sensory feedback, change the gain of that feedback, and essentially mimic the presence or the absence of these inhibitory neurons that we eliminated. And so when he increased the gain and essentially got rid of this inhibition, what we found is that there was this characteristic damped harmonic oscillation that very closely resembled what we found in the mouse. And so what we think we've begun to uncover by combining this high-resolution behavioral assay with genetic tools and modeling tools is an inhibitory microcircuit in the spinal cord that's really dedicated to maintaining the stability of the limb as we reach for a target. Now I want to get to this other idea that I brought up, this idea of internal copies, so something that can compensate for these sensory feedback delays and allow us to make very rapid corrections. So again, the idea is copies of motor commands are conveyed to the cerebellum. The cerebellum can make these online predictions of the outcome of those commands. But this idea is, again, remain largely theoretical because it's been very hard using traditional approaches like electrical stimulation to modify just the copy. When you stimulate, you tend to perturb the copy and the command simultaneously, since by definition these copy branches branch off of these command pathways. So what I wanted to do is see if we could use mouse genetics to gain selective access to a copy pathway and really ask a question that couldn't be answered before. Does an internal copy pathway actually have any contribution to motor behavior? So I looked at this specific class of neurons in the cervical spinal cord that controls forelimb muscle contraction, these propriospinal neurons, and they have two features that are very relevant in this scheme. So one, they receive a lot of descending motor command information from the brain, and two, they have this very characteristic bifurcating output. One branch of these neurons descends and projects to the motor neurons that innervate our forelimb muscles and cause these muscles to contract. And the other branch from these same neurons ascends into the brainstem to this structure called the lateral reticular nucleus, or the LRN. It doesn't really matter all the acronyms, but the LRN is a major input to the cerebellum. So at face value, it looks like these neurons are very well equipped to convey motor commands to the forelimb and copies of these commands up to cerebellar circuits. But again, does this internal copy branch actually have any function, any contribution to motor behavior? So using similar genetic methods in the mouse, I found a way to access these neurons within a class of genetically identified neurons in the spinal cord. Again, use these viral tricks to eliminate these neurons acutely in the adult mouse, and then looked at the behavior. And this mouse also has quite a bit of trouble. Essentially, he becomes dysmetric. In this case, what that means is he's underreaching the target. Sometimes they overreach. He's going to try another motor strategy in a second. Right there. And so essentially the trajectory is this meandering on the way to the target. It takes much longer for them to reach. Their success drops down to 0. And I think one of the most striking things is how different this phenotype is from what we saw when we eliminated those other neurons. So what this really indicates is you can get into the spinal cord and use these genetic tricks to disentangle discrete classes of neurons and actually define very different contributions to the same behavior. But that leaves open the question that I've been raising a couple times. So when you kill a neuron, of course, you eliminate both of its outputs. So does this branch have any contribution to motor output? So I wanted to test that, and to try to get selective access just to this branch, I decided to use an optogenetic approach. And so fortunately, you just heard what channel rhodopsin is, so I won't introduce it again. But what I did is I expressed channel rhodopsin, this light-gated ion channel, into these propriospinal neurons. And the key to this experiment is I introduced light. I photostimulated only at the axon terminals up here in the LRN. So what that allowed me to do is activate this internal copy pathway. So here I'm recording in the LRN while I photostimulate these PN axons, and you can very robustly activate neurons in this structure, and these are the neurons that are going to project to the cerebellum. And in data I'm not showing here, we found that you only activate this branch, the rest of the neurons unaffected. So you can really manipulate the internal copy branch and leave the premotor branch behaving normally during a reaching task. So now we do the reaching task again, and here I'm photostimulating, imposing this synchronous activation at 20 hertz, and every time I photostimulate, you see this severe perturbation of the limb, the severe muscle contraction phenotype. And you can see that this is very robust. These blue traces are the reaches with the photostimulation, and they essentially just have this big perturbation of their limb every time I photostimulate. So this indicates that this internal copy pathway can have actually a very strong and robust effect on motor neuron activity. And in a final set of experiments, we are beginning to actually figure out how it does this. So we've identified this very rapid feedback loop that passes through the cerebellum and gets back to motor neurons in about 5 milliseconds. So stepping back, in the context of a reaching movement that takes maybe 200 or 300 milliseconds, you can imagine an internal feedback loop that passes through the cerebellum and can modulate motor neuron activity every 5 milliseconds is well equipped to update motor neuron activity, correct motor neuron output, and really refine the trajectory continuously throughout a behavior. So in ongoing work, what I'm doing now is trying to combine these behavioral approaches with some of the imaging approaches and electrophysiological recording approaches that you just heard. So can we quantify the activity of these internal copy circuits during behavior and actually get a sense of the type of information that they convey during a reaching movement? So basically, to sum up, what I've been describing are our efforts to combine, to combine these genetic, electrophysiological, recording, we're pursuing imaging experiments, behavioral and theoretical approaches to get a better sense of how the motor system solves key challenges in motor control. So all of these approaches generate a great deal of data. So if anybody has any ideas on the better ways to analyze the masses of behavioral data we're getting and how we combine that with the imaging data we're getting, I'm all ears. But the challenge is essentially to continue to explore these circuits, bring the behavioral precision of primate studies into the realm of mouse genetics, where we can take a reductionist approach and dismantle the neural circuits really one element at a time and get a better sense of how mice, and I think by extension primates, control these skilled movements.
So based on your expert opinion, what's more difficult and why? Moving really fast or moving really slow?
Difficult in what respect? As far as achieving a goal?
Up to you. Up to you. How do you find difficult?
I mean, I can move very quickly if my goal isn't difficult to achieve, and I have no problem moving quickly. I might fall over, so I'm not really sure what the goal is.
Okay, sure. Let me just add some more context.
Yeah.
So like moving really slowly would be like doing Tai Chi, and moving really fast would be like the chopstick video you showed earlier.
Yeah.
So of those two examples, which one do you think is more difficult and why?
I can't do either, so I don't know.
I can't do either. That's why I'm asking, really.
I think they're going to recruit very different. They're very different challenges for the motor system. One, the first one is really what I've been stressing. How do you make very rapid corrections to refine trajectory? The other one is really a matter, I think, of overcoming muscle fatigue, maintaining stability, maintaining co-contraction of your limbs so that you can maintain both postural stability and limb stability. So there are different challenges, but which one's harder? I don't know. I'd have to try them both, I guess. I'll get back to you on that.
Here. I may miss some detail. How much of the antidromic stimulation of PN contribute to the defect of the motion?
So when we photostimulate the PNs.
Right.
So yeah, so I went through this briefly, but essentially what we found, which isn't the case in every one of these photostimulation of axon terminal experiments, we looked really carefully through a number of methods. I won't go into all the details, but we found no evidence that when you photostimulate here and activate this pathway that there's any antidromic stimulation. We recorded for many of these neurons, never saw any antidromic spikes. We lesioned the connections from the LRN to the cerebellum and saw that most of that motor effect was eliminated. But in you have to be very careful and test this in every circuit that you want to try this in, because in many other circuits you do get robust antidromic activity. It's really a circuit phenomenon, I think.
Oh, great, thanks.
I'm thinking about what you've suggested, that this system is primarily concerned with fast correction of motor actions, but I wanted to propose that alternatively you could see the PN information that is going up to the cerebellum is providing the context for the ongoing motion. So basically it's a necessary component for the healthy reaching, if you like.
So we have some evidence that, so for example, there's a lot of cerebellar lesion experiments in patients with cerebellar lesions, and you don't need this, the lesions of course are fairly nonspecific, but you don't need a cerebellar pathway to initiate and generate movements. It's important for the coordination, the refinement, the adaptation of these movements.
That's why I meant continuation. I mean, so if you start it with a motor command and you need the context to continue producing it, so say, you start reaching it, and even though it's like, I don't know, 20, 30 milliseconds, during these 20, 30 milliseconds, if this loop is really 5 milliseconds long, it is producing, a couple of iterations of that, carry on reaching, so to speak.
Yeah, that feedback could be essential for maintaining enough excitability in the system so that you can continue your movement. I think from what we know, it's most likely that this feedback is essential for tweaking and refining, and then on a longer time scale, very interestingly, it's essential for our adaptation to new environments. So our muscles fatigue when we're moving. If you put a bat in my hand and I have to adapt my model of this sensory motor apparatus to this new bat that's in my hand. And so it's thought that these copy pathways are also important on longer time scales, because then you can compare what you think is going to happen with what's actually happening. You get better trial after trial. So I think on every time scale, the short time scales you're mentioning and long time scales, they're going to be contributing to motor output. So I just want to thank everybody involved, my PI, especially Tom Jessel, who's been really an amazing mentor throughout my postdoc. I want to point out Andrew Fink, especially, who is a former graduate student in the lab, very talented former graduate student who initiated the GABA project. He's now a postdoc in Richard Axel's lab. And of course, I want to thank Larry, who developed the model that I was able to describe briefly. My collaborators on the PN project, Bor Alstermark and Zhuan Zhang, all the members of my lab, especially Katie, a really diligent technician. And I want to thank a few people, John Krakauer, Alla Karpova, and Mark Churchland, who've provided a lot of conceptual and methodological advice throughout my postdoc. And finally, these are the funding sources that made the work possible.